Okay, hello everyone, my name is Katie, and today I will be talking to you about gastropods. So, class Gastropoda is a major molluscan lineage encompassing a wide range of familiar forms, which include snails, slugs, conchs, whelks, abalone, and limpets. In general usage, the term snail refers to gastropods bearing a large external shell into which the body can fully retract, whereas slugs lack an external shell or retain only a highly reduced internal remnant. Gastropods live in a wide variety of habitats, but marine gastropods display particularly striking morphological diversity, with many groups such as abalones, cowries, periwinkles, and whelks possessing tightly coiled adult shells. In contrast, limpets and related taxa exhibit shell coiling only during the larval stage and retain a simple conical shell form as adults. Gastropods first appear in the fossil record during the Cambrian period, approximately 540 million years ago. This establishes them as one of the oldest continuously successful animal lineages. Their long evolutionary history is marked by repeated ecological expansion across marine, freshwater, and terrestrial environments. The clade is defined by several key anatomical and developmental features. These include a univalve shell, when present, an operculum borne on the foot, and a well developed head with concentrated sensory structures such as eyes, which reflects pronounced cephalization. Most importantly, gastropods are uniquely characterized by the process of torsion, which I will talk about later on. This is the 180-degree rotation of the visceral mass during larval development that reorganizes the body plan and represents the diagnostic apomorphy of the group. For little etymology, the term gastropoda means stomach foot, and the foot refers to their ventral muscular foot. So this slide helps situate gastropods within the broader clades we've been discussing throughout the lectures this semester. Zooming in towards the base of the tree, hopefully you can see my little laser pointer here. Gastropods are placed within the phylum Mollusca. This is a group defined by a characteristic body plan that includes a muscular foot, mantle, visceral mass, and ancestrally a radula and mineralized shell. Within Mollusca, gastropods belong to Conchifera, the shell-bearing mollusks, and are distinguished from aculiferans by inheriting a single mineralized shell and mantle cavity. So the placement of gastropods within mollusca has been shaped by a long history of competing phylogenetic hypotheses derived from morphological, developmental, fossil, and most recently, molecular data. Much of the debate has centered not on gastropods themselves, which are consistently recognized as a coherent lineage, but rather on how the major molluscan classes relate to one another, particularly the positions of the aplacophorans, monoplacophorans, scaphopods, and cephalopods. These uncertainties produce several alternative higher-level frameworks for molluscan relationships, which are very nicely summarized in this figure right here. And as you can see, some of these frameworks don't even explicitly list the gastropods. The three major hypotheses historically dominated discussions. Most controversy centers on the monophyly of Aplacophora, the relationships within Conchifera, and the placement of Polyplacophora. So, for instance, is this in the clade Aculifera, or is it in Testaria? So the first is the Serialia hypothesis. This unites chitons, the Polyplacophora, and monoplacophorans based on serial repetition of organs, and then it places all remaining mollusks, including gastropods, into a separate clade. Next is the Aculifera and Conchifera hypothesis, which propose that the chitons and the aplacophorans form a clade right over here, Aculifera, and this is sister to all other mollusks which are grouped under Conchifera. The Testaria or Eumollusca hypothesis treats Aplacophora as a basal paraphyletic clade, with chitons grouping all other shelled mollusks over here down in the clade Testaria. The modern phylogenetic consensus is supported primarily by large-scale molecular and phylogenomic analysis, and it strongly favors the Aculifera-Conchifera dichotomy. So under this framework, living mollusks are divided into two major clades, Aculifera here in pink. This includes the shellless aplacophorans and the Polyplacophora. They are united by features such as calcium carbonate spicule and epidermal papillae. Conchifera here in green. By contrast, includes the monoplacophora, cephalopods, scaphopods, bivalvia, and of course the gastropods. They are all characterized ancestrally by the presence of a single or paired calcified shell, which covers the dorsal surface of the body. Within the current phylogenetic framework, gastropods are very securely placed as a major lineage within Conchifera. While this higher level placement is no longer controversial, some uncertainty remains regarding the exact position of Gastropoda within the clade. For example, over here in Figure B, a genome scale analysis by Chen et al. In 2025, they recover Gastropoda within this clade right here, which is sister to the clade comprising of bivalvia and Scaphopoda. On the other hand, morphological analysis summarized by Dominova and Zukov in 2022, they place gastropods as more closely related to the cephalopods, and then bivalves diverge earlier over here within the conchiferid lineage. So now we zoom in from mollusca to gastropoda. So for most of the 20th century, gastropods were divided into three major subclasses: Prosobranchia, Opistobranchia, and Pulmonata. Starting in the 1980s, however, studies of microanatomy and genetics indicated that most of these were not monophyletic groups and that new arrangements were needed. So the following 6 major clades of living gastropods are now generally accepted. So 1, 2, 3, 4, 5, 6. And I'm going to briefly discuss the features of each in later slides. So on screen is one phylogenetic hypothesis for these major clades developed by Uribe et al. In 2019. Note that Caenogastropoda and Heterobranchia, they form a clade, this thing in blue right here, known as Apogastropoda. Collectively, Apogastropoda, Nertomorpha, Vetagastropoda, and Nymphaliina, so in other words, all of the extant subclasses except Patelogastropoda, they form this clade right here in green, known as Orthogastropoda. Lastly, down here in gray, you have Patelogastropoda, or the true limpets. This is the sister group to Orthogastropoda and the basalmost clade. So despite major advances in molecular systematics, the relationships amongst the major gastropod clades remain unsolved, largely because different analytical approaches recover conflicting topologies. So the 6 major gastropod lineages tend to shift positions depending on the type of data analyzed. So for instance, here in early cladistic analysis based on morphology, so for instance, the one by Ponder and Lindbergh in 1997, they tend to consistently recover Patelogastropoda as the sister group to all other gastropods, and they form the clade Eogastropoda. So this framework was very influential for decades and strongly shaped traditional views of gastropod evolution. However, come the 2000s, early molecular phylogenetic studies produced inconsistent results regarding the placement of Patelogastropoda. This raised doubts about the stability of Orthogastropoda. So more recent analyses using large nuclear datasets have either supported the deep split between Patelogastropoda and Orthogastropoda, as recovered here by Kokot et al. In 2011. But some also favored a sister group relationship between Patelogastropoda and Vetagastropoda, thereby rejecting Orthogastropoda as a monophyletic group. So in contrast, mitochondrial genome-based phylogenies have consistently recovered a different pattern. They tend to place Patelogastropoda as sister to Heterobranchia. So this topology challenges the monophyly of both Orthogastropoda and Apogastropoda and highlights the strong influence of the mitochondrial signal, but the paper does note that this may be affected by factors such as lineage-specific rate variation and long branch attraction LBA artifacts. So in short, Gastropoda is firmly monophyletic, but resolving the relationships between the major clades to be congruent with morphological and genomic datasets is still an area of active research. Okay, so to talk a little bit more about the 6 different subclasses, and I'm not going to talk too much about the cladograms because if I did, this presentation would be about an hour. But anyways, so first off is Caenogastropoda. This is the most diverse gastropod clade, comprising about 157 extant families, 190 extinct families, and roughly 60% of living snail species. Most possess a shell that lacks a nacreous mother-of-pearl layer, and they have a single monopectinate cnidium, which refers to a gill that consists of only one structure, with filaments arranged on only one side of a central axis. All Canogastropoda share an orthostrophic protoconch in which the larval shell is coiled in the same orientation as the adult, but this delicate feature is rarely preserved in fossils and also occurs in Nertamorpha. Next, we've got Heterobranchia. Very aside, but I think this is the coolest looking clade, so therefore they get 2 slides. But anyway, so Heterobranchia includes 2 major ecological radiations. You've got the marine sea slugs and bubble snails, formerly grouped under Opistobranchia, and the majority of land snails and all terrestrial slugs, formerly grouped under pulmonates. Heterobranch shells, when present, always lack nacre and are highly variable in form. So in many lineages, the shell is reduced, internalized, or completely lost altogether. Limpet-shaped shells have evolved multiple times independently within the group, illustrating extensive morphological convergence, and most Heterobranchia lack an operculum. Their protoconchs are typically sinistral, meaning that the larval shell coils to the left, whereas adult shells often exist heterostrophy, in which the orientation of the adult shell differs from that of the larval. Modern phylogenetic work recognizes 2 major Heterobranchia assemblages. So we got the so-called lower Heterobranchia. They represent a paraphyletic clade of mostly marine basal lineages. The remaining form Euthenera, a largely monophyletic clade, which includes Actionacea, the small-shelled marine snails, Nudipleura, the small shellless or reduced shell sea slugs. These also includes nudibranches, and Tectipleura, which unites the Eupistobranchia, which includes the sea hares, theropods, bubble snails, with Panpulmonata. This includes the land snails and slugs. Next is Nertamorpha. This is characterized by a mosaic of plesiomorphic and apomorphic features, a combination that has contributed to long-standing uncertainty regarding its phylogenetic position within Gastropoda. Although less speciose than Canogastropoda or Heterobranchia, the clade includes approximately 2,000 extant species, including over 500 terrestrial species. They typically possess small, non-nacreous shells ranging from coiled to limpet-shaped forms, often with teeth or folds on the inner lip of the aperture, and they are further distinguished by an outer homogeneous calcite layer. Vetigastropoda is an exclusively marine clade, and their defining characteristic is the presence of a nacreous shell layer, which often produces a very pretty iridescent sheen. This feature is considered a retained plesiomorphy within mollusca and links vetigastropods more closely to other nacre-bearing conchiferans. In coiled vetigastropods, a circular and heavily calcified operculum is typically present, whereas taxa with limpet or abalone-like shell morphologies have secondarily lost the operculum. Neomphalina is a relatively obscure but evolutionary significant clade of deep-sea specialists associated primarily with hydrothermal vents. They exhibit extreme adaptations to these habitats, including hypertrophied gills and close symbioses with chemoautotrophic bacteria, which often forms the basis of their nutrition, rather than grazing or predation. One species I do want to take the time to highlight is Chrysomalin squamiferum, which is shown on screen. This is also known as the scaly-foot snail, and it was discovered at Indian Ocean hydrothermal vents. So they are unique amongst animals in that they incorporate iron sulfide minerals, sometimes referred to as fool's gold, into both the outer layer of its shell as well as the sclerites. These are the little sclerites, the spiky bits extending from its foot. This represents an unprecedented form of biomineralization and really highlights Neomphalina as an important model for studying evolutionary innovation under extreme environmental constraints. The scaly-foot snail also caused the clade to get a lot of media attention following its discovery. Thought this was kind of fun. I highlighted a few articles over here. Lastly, we've got Patelogastropoda. They comprise the true limpets. They are a relatively species-poor but morphologically distinctive clade, adapted primarily to wave-exposed, rocky intertidal habitats. They are characterized by uncoiled, cap-shaped shells and a powerful muscular foot, which enables strong adhesion to rocks under high hydrodynamic stress. Because of the vast diversity we have just seen, gastropods exhibit really remarkable anatomical and ecological variation, but this also makes broad generalizations difficult. That being said, gastropods are united by a single shared developmental process, torsion, which therefore serves as the diagnostic apomorphy of the clade. Gastropods begin development with the mantle cavity, gills, sensory structures, and anus positioned posteriorly, similar to the generalized molluscan condition. So this end posterior, this end is anterior. During the veliger larval stage, the visceral mass undergoes a counterclockwise rotation of up to 180 degrees. This repositions the mantle cavity and the anus to be anterior above the head. As a result, the gut, so this is the green right here, becomes U-shape and the nervous system is reorganized. So prior to torsion, the gastropod nervous system is eutheneural, with the paired visceral nerves running parallel along the body. But torsion transforms this arrangement into a streptoneural condition in which the nerve cords cross in a characteristic figure 8 pattern. This is a condition known as chiastoneury. The foot itself remains unaffected by torsion, retaining its original orientation and function. The adaptive significance of torsion remains debated, but a leading hypothesis emphasizes defense, where anterior placement of the mantle cavity allows the head to retract first, followed by the operculum, which therefore seals the shell opening and increases protection from predators. I will note that in some heterobranch lineages, torsion is followed by detorsion, which is the secondary and partial reversal caused by differential growth rather than true mechanical twisting. Crucially, even in these detorted groups, torsion is still homologous across Gastropoda, as it occurs during development in all members of the clade. Again, that's why it's the diagnostic apomorphy. So most shelled gastropods possess a single one-piece shell, typically coiled or spiraled, at least during the larval stage, with the aperture usually opening on the right-hand side. So this means the shell is dextral. So this right here is a dextral shell. So when you're viewing the shell with the apex oriented upwards, you can see here it opens on the right. I do want to highlight this over here is an example of a sinistral shell. So again, apex pointing upwards, shell opens here on the left. This is what we talked about in the previous slides with the heterobranchia protoconchs. Anyways, so ancestrally, gastropods have a univalve shell, but this condition is not unique to the clade. Monoplacophorans and several other extinct molluscan groups also possessed single shells. So this univalve condition represents a plesiomorphic trait inherited from ancestral conchifera, and while it does not constitute a true diagnostic apomorphy, it is seen as a clade-defining feature. But there are a few highly specialized gastropod lineages that have independently evolved a bivalve shell morphology that superficially resembles that of true bivalves. The most notable example is the family Julidae, in which the ancestral univalve shell becomes developmentally divided into two articulated valves. This represents a clear case of convergent evolution, as the bivalve condition arises independently and does not reflect shared ancestry with bivalvia. So despite their clam-like appearances, so here's one example, it's pretty cute in my opinion, but these gastropods retain key synapomorphies, including torsion and a functional radula. This confirms their placement within Gastropoda rather than bivalvia. Okay, so we can now turn to why this clade matters. Why am I even talking about this? Well, for starters, gastropods constitute the most diverse lineage of extant mollusks, accounting for approximately 80% of all molluscan species and more than 70,000 living species. This exceptional diversity is tightly linked to their ability to colonize an unparalleled range of environments, which includes marine habitats at all depths, brackish and freshwater systems, fully terrestrial ecosystems, and arboreal niches. Notably, gastropods also inhabit some of the most physically and chemically extreme environments on Earth, including hydrothermal vents, which we talked about earlier, and this highlights their extraordinary evolutionary and physiological adaptability. So this is just a figure that I took from the Paleobiology Database Navigator, and it denotes over 167,000 documented gastropod occurrences worldwide, spanning the entire Phanerozoic eon. And so I think that this visualization is really neat in that it highlights the extraordinary diversity and long-term evolutionary persistence of gastropods. This is another PBD image. So to briefly touch on time scales, the Cenozoic era, so this is the yellow right here, the text was just a little too small to fit. But the Cenozoic era stands out as a period of major radiation, particularly at the genus and species level for gastropods. It is during this time that a lot of the modern diversity became established. And this diversification is closely tied to the ecological release following the end Cretaceous mass extinction, which created many vacant niches, especially in shallow marine environments into which gastropods rapidly occupied. Okay, so there are many reasons to study gastropods, but one that I find particularly compelling is, again, their dominance within deep-sea hydrothermal vent ecosystems, especially amongst members of the clade Nymphaliina. These environments act as natural laboratories for examining how life evolves under extreme physical and chemical constraints, which really offers insights into the limits of metazoan adaptation. As discussed earlier, species such as the scaly-foot snail over here, they inhabit low-temperature, diffuse flow zones along Indian Ocean ridge systems and often constitute a really substantial fraction of total hydrothermal vent biomass. Their extreme morphological and physiological specializations make vent-associated gastropods powerful model systems for studying adaptation to high temperature, toxic, and hypoxic environments. As a brief side, I'm particularly very interested in the origins of life and astrobiology, which is one of the reasons I took this course. And so these systems are especially compelling to me because they may approximate conditions on early Earth and potentially extraterrestrial environments. So in that sense, vent-associated gastropods highlight the broader relevance of this clade, not only to evolutionary biology, but also to astrobiology, and I think that is very neat. Okay, so we have made it to the end of the presentation. Thank you very much for listening. I hope you enjoyed hearing about gastropods. I certainly enjoyed learning about them. And thank you again for a really amazing semester.